So RNA is mostly produced in IVT, and in that process, it can be either capped, so it must be capped to be functional, in the IVT process, or as a secondary enzymatic reaction after the original IVT is completed. And that enzymatic reaction produces both capped, uncapped, and intermediate species, where co-capping produces capped and uncapped RNA with no intermediates. And then, of course, we get analytics at the end to identify how much of the RNA has been successfully capped. So most current cGMP release assays use a 5' drop-off assay. So that might be DNazyme, it might be RNase-H-based, but there's an oligo that cleaves the front end of the RNA off, the rest of it is discarded, and then HPLC separation of those small fragments can help identify which one kind of cap and which one is not. But that requires sequence-specific optimization and HPLC optimization to make sure that you're separating things and seeing what you need to see. And that can be timeline and cost prohibited in the RG space, which is where, of course, a lot of our customers begin. So that brings me to Endeavor's VaxArray 5' CapQ assay. This is a multiplexing and plate-based or slide-based technique to measure various analytes. It can do a lot more than the 5' CapQ, and so I would advise you to speak to Randy Lindsay and Barry Costa, who are here. They are experts in the instrument, I'm not. But we are using it for the 5' CapQ assay, which measures how much RNA is capped. So there's a diagram here from Endeavor about how this works. There's a capture antibody that binds to the 5' cap. So only RNA binding to the plate will have the cap. Anything uncapped will not bind. And then you have a fluorescent oligo that binds to the poly A tail. So a complete functional RNA must have the cap and must have the tail. And therefore only those complete functional RNAs will bind and then fluoresce. So only those will be measured and read out in this assay. And then I've got some of the calculations down there on the left, but you can then use this measure of intactness of your RNA that you've done ahead of time to estimate how much of it you think is that full length, and then use that to estimate how much of it is capped. So it's a measure of capped and intact RNA. So go with me on this, but this is a graph of the types of RNAs that are possible with enzymatic and co-capping reactions. This first one is the only species that will perform as expected in a cell or in a human. It's got the cap, it's got the tail, and it's full length. It's coding for what you hope it codes for. The second one is an example of an RNA that was made complete but did not get the cap. The third one is degradation or truncation, incomplete transcription perhaps, where there's a cap but no tail. Then we can see 5' truncation, no cap, yes tail. And then something that is truncated, but if it was enzymatically capped, could have both cap and tail, but not in full length. Last, we could see truncation of both ends of the RNA, so it's small and does not have a tail. And if you're enzymatically capping, you could take some both ends truncated species and also cap them. So all of these species are possible and likely to be present at varying percentages in your final product, although of course we hope it's as much as possible in the first one. So a 5' drop-off assay will only measure things that do have the expected structure of the 5' end. So anything with degradation to the 5' end will not be seen by that assay. So when it measures percent capped, it measures the amount that does have the cap, but does not care if it has a tail or not, over everything that had the entire 5' end. So I've boxed the numerator in orange and the denominator in red for what that assay would see and measure. The pros of this are that it directly measures uncapped instead of deducing it as a balance of what isn't capped. It's very quantitative, and it can, if properly optimized, distinguish cap intermediates if you've gotten there enzymatically. But the cons are that many of those other species that include 5' degradation are not measured at all in such an assay. So then if we look at the 5' CapQ assay, because you are using an orthogonal measurement of full-length product, all of the species that contribute to the mass of your RNA are present in the denominator. Everything is looked at as being potentially available, and then the only things that are measured in the assay and are counted as capped and intact are anything that has a tail and has a cap, which is primarily that first species, which is the species of interest. That other species where there's 5' truncation but has been enzymatically capped wouldn't be possible in a co-capping reaction and is expected to be a very minor presence even in enzymatically capped. So the pros of this approach are that it sees more of the relevant species and therefore gives you a bit of an orthogonal measure of what your final product actually looks like when used in conjunction with HPLC measurement. And the output is the relevant measure of what is capped and sealed, so that's biologically relevant. The cons are that it does require that orthogonal measurement of FLP, so you need to know how intact your RNA is going in. And it can only be quantitative if you do have something that you already know how capped it is to use as standard curve. And it cannot distinguish enzymatic cap intermediates, although since most clinical products are using co-capping, that's less of a concern. So we at Aldevron in the R&D department have been using this across a wide range of constructs because it doesn't require as much construct-specific development. So rather than having to sequence-specific design all of those, we can use this for any product of any length. So from left to right, we have a small reporter gene, about a kilobase. In this case, it was a GFP. Then we have something more in the mid-range. This is about the size of your base editors, like a Cas9. And then our 6 kilobase construct is a relatively short self-amplifying RNA. So for any of these, we, with just some small standard curve optimization, we're able to get good signal and good saturation, and therefore report on the percentage of cap without having to do extensive sequence-specific design. So as you would expect, the longer your construct is, the lower the molarity is the solution, and that can affect signal. But with these standard curve designs early on, it's easy to figure out how much you need to load, what that looks like. And it's a fast lift, especially compared to the months of development in HPLC optimization that you might see with a different approach. So, in practice, we've used this to help improve enzymatic capping processes. So in this experiment, we began with an enzymatic process that was not optimized, and it included relatively high pH and relatively high concentrations of magnesium, which were necessary to make the enzymes do their job, but degraded RNA. So in our original condition on the right, I'm first presenting HPLC capping information, so that did distinguish cap 1 and cap 0. And then on the left-hand set of bars, red and green, is cap 1 and cap 0 after optimization. So we did a DOE, identified improved conditions that changed pH, magnesium, and several other buffer components, and then used a 5' drop-off HPLC assay to estimate how much was capped. You can see that in both cases, the red and the green bars sum to about 99% capped, which is wonderful. But in the case of the optimized condition, we were able to move about 6% of the cap 0, which is a sufficient cap product, but not as good as a cap 1, into that cap 1 bar. So we not only minimized degradation, but also improved how much of the material had cap 1. Then I've added here the data from the 5' cap Q VaxArray assay. That's in blue. Furthest left bar to the standard curve, and then the rest of the data is what's been seen before, but with the addition of the VaxArray data. So in this case, the optimized condition, so the blue bars should be equivalent to the red and the green combined, since the VaxArray did not distinguish those two species. In the optimized condition, we see exactly what we saw with HPLC, so that was nice to have that confirmed, 99 to 100% of performance. However, in the original condition, we were seeing 99% cap with HPLC, but only getting 62% capped and intact with VaxArray. And our hypothesis is that this is reflecting the fact that those assays see different populations of RNA. So anything that has been degraded is more likely to be read by the VaxArray, less likely to be read by the HPLC, and this confirms our need to optimize. So these two sources of data combined are giving us information about how intact the RNA is, how functional it will be, and how much of it is the expected species. So the last thing I'm going to touch on today is co-capping with ARCA. ARCA is the anti-reverse cap analog. It is at this point, sort of elder statesman of the cap analog world, which also means that it is remarkably cheap. And the struggle with this cap analog is to get good yields and high capping efficiency. So Aldevron has 2 T7 polymerases that we use to make RNA. We've got our wild-type T7 that we've been using for a long time, and in the past few years, we've rolled out the Codex high-cap T7, which has the advantages of using less cap analog and minimizing double-stranded RNA. Another strength of this is that it does not need to then GTP start when using ARCA. So ARCA reactions are cheap for cap analog, but we need to minimize the amount of GTP in the reaction in order to get the polymerase to use the ARCA co-cap. And then GTP becomes a limiting reaction and we get less material out of the reaction. So in this experiment, we varied the amount of ARCA in the reaction at either 1.5 or 4 millimolar. And the red squares and circles are our wild-type reactions. So when ARCA was relatively high in the reaction and GTP was low, we got decent capping efficiencies, about 60, that's red squares, but relatively low yields. And then when we minimize ARCA and increase GTP, we got better yields, that's red circles, but capping efficiency went down the drain. It was very, very low. However, with high cap, we did not need to starve GTP, so in both cases, the reactions had plenty of materials. Yields were great, that's our blue squares and blue circles, and there was excellent capping efficiency at both 1.5 and 4 millimolar of ARCA. So great capping efficiency and great yields, and all the data here for capping efficiency were VaxArray related because this was a co-capping process, so there was no question about intermediate species.